Here we see how ATP synthase makes ATP. Protons flow down their electrochemical gradient through the stationary stator. Each proton binds to a subunit of the rotor, causing it to change shape and makes the rotor spin. The spinning rotor turns the connecting rod. As the rod spins, it activates catalytic sites in the knob that join inorganic phosphate to ADP, making ATP. This atomic force microscopy shows the structure of the ATP synthase molecules. The ATP synthase works like an ion pump in reverse. The proton gradient also drives energy need for ADP, pyruvic acid, inorganic phosphate, and calcium to cross into the inner mitochondrial membrane. Here is a brief animation about the ETC. Notice how electrons are delivered by NADH and FADH2, and how these electrons move through the electron transport chain complexes. The last complex adds a pair of electrons to oxygen to form water. The energy from the electron movements pump protons into the inner membrane space, establishing the proton gradient. Lastly, the protons move down the proton gradient through ATP synthase, and ATP is formed. Cyanide, the gas used in gas chambers, disrupts oxidative phosphorylation by binding to cytochrome oxidase. This blocks the electron flow from complex 4 to oxygen. Poisons called upholders abolish the proton gradient by making the inner mitochondrial membrane permeable to protons. Although the ETC continues to deliver electrons to oxygen at a furious pace, the oxygen consumption rises, and no ATP is made. To summarize, energy use at rest averages 100 kilocalories per hour, so this places a huge demand on mitochondria for energy production. Of the 686 kcals in 1 mole of glucose, about 262 kcals are captured in ATP, with the rest lost as heat. This translates as a 38% efficiency in usage of fuel available. Machines are only around 10 to 30% efficient. The flow of energy is as follows. Ingested glucose is converted to NADH plus H plus, which goes through the ETC, creating a proton gradient, which makes ATP. the net gain from the complete oxidation of one glucose molecule. In substrate level phosphorylation, we make 4 ATPs, 2 from glycolysis and 2 from citric acid cycle. In oxidative phosphorylation, we make 28 ATPs. For the NADH plus H plus brought in, the proton gradient generates 2.5 ATPs. 10 NADH plus H pluses are made, so 25 ATPs. For every FADH2 brought in, only 1.5 ATPs are created. Since we make 2 FADH2s, we make a total of 3 ATPs via that route. The total ATP made from substrate level phosphorylation and oxidative phosphorylation is 32 ATPs. But remember, 2 ATPs are required to move the NADH plus H plus into the mitochondria for glycolysis. So the net energy made from one molecule of glucose is 30 ATP. There is still some uncertainty on the final total. However, for our class, we will be using 30 ATP as the final net energy made. Figure 24.11 shows the summary of the energy yield from cellular respiration. During glycolysis, 2 net ATP are made via the substrate level phosphorylation. From the Krebs cycle, 2 more ATP are produced. Also, via the substrate level phosphorylation and from the electron transport chain, about 28 ATP are made via the substrate level phosphorylation. This totals 30 ATP made per glucose molecule. This is a short video that explains how we make ATP energy from ingested food that has been broken down. This is a great overall of the whole process. Whoops.
As this mountain biker heads up the trail, the breakfast he ate this morning is being burned to power his bike ride. His breathing rate increases as his leg muscles demand more oxygen to burn more fuel. Let's zoom down to where this fuel is burned, our cells. Here, the blood vessel on the left delivers fuel and oxygen to a single muscle cell. In cellular respiration, energy and fuel is converted to ATP, shown here as starbursts. Most ATP is made in the cell's mitochondria. ATP powers the work of the cell, such as contraction. Let's take a closer look at how ATP is produced from a molecule of glucose, our fuel. Only the carbon skeleton is shown to keep things simple. The first step is called glycolysis, and it takes place outside the mitochondria. To begin the process, some energy has to be invested. Next, the molecule is split in half. Now, the molecule NAD+, an electron carrier, picks up electrons and hydrogen atoms from the carbon molecule, becoming NADH. Keep track of the electron carriers. They play an important role by transporting electrons to reactions in the mitochondria. In the final steps of glycolysis, some ATP is produced, but not much. For every glucose molecule, only 2 net ATPs are produced outside the mitochondria. However, glycolysis has produced pyruvic acid, which still has a lot of energy available. Let's follow this pyruvic acid molecule into a mitochondria to see where most of the energy is extracted. As the molecule enters the mitochondria, 1 carbon is removed, forming carbon dioxide as a byproduct. Electrons are stripped, forming NADH. Coenzyme A attaches to the 2-carbon fragment, forming acetyl-CoA. Coenzyme A is removed, and the remaining 2-carbon skeleton is attached to an existing 4-carbon molecule that serves as the starting point for the citric acid cycle. The new 6-carbon chain is partially broken down, releasing carbon dioxide. Several electrons are captured by electron carriers, and more carbon dioxide is released. The carbon dioxide that you exhale comes from the reactions of cellular respiration. 2 ATPs are produced by the citric acid cycle for each molecule of glucose. At this point, only a small number of ATPs have been produced. However, more energy is available in the electrons that are being transported by electron carriers. While the citric acid cycle starts another round, let's follow an electron carrier to the next step in the process. Electron carriers such as NADH deliver their electrons to an electron transport chain embedded in the inner membrane of the mitochondria. The chain consists of a series of electron carriers, most of which are proteins that exist in large complexes. Electrons are transferred from one electron carrier to the next in the electron transport chain. Let's take a closer look at the path electrons take through the chain. As electrons move along each step of the chain, they give up a bit of energy. The oxygen you breathe pulls electrons from the transport chain, and water is formed as a byproduct. The energy released by electrons is used to pump hydrogen ions, the blue balls, across the inner membrane of the mitochondria, creating an area of high hydrogen ion concentration. Hydrogen ions flow back across the membrane through a turbine. Much like water through a dam, the flow of hydrogen ions spins the turbine, which activates the production of ATP. These spinning turbines in your cells produce most of the ATP that is generated from the food you eat. The process you just observed, cellular respiration, generates 10 million ATP per second in just one cell. That ATP can power a biker up the trail, or it can power your brain cells as you learn challenging biology topics.
Cells cannot store large amounts of ATP. Rising intracellular levels of ATP inhibit glucose catabolism and promote glycogen or fat formation. Glycogenesis is the process of making glycogen to store the excess glucose. Glycogenesis is catalyzed by the glycogen synthase. Glucose is converted to glucose 6-phosphate, then converted to the isomer glucose 1-phosphate. The terminal phosphate is removed as the enzyme catalyzed the attachment of glucose to the growing change. Glycogenesis mostly occurs in the liver and in skeletal muscle cells. Glycogenolysis is the breakdown of glycogen via glycogen phosphorylase into glucose in response to low blood sugar. So this is taking the storage form glycogen and reversing it into glucose for cell use. The enzyme splits and phosphorylates the terminal glucose and glycogen. It forms glucose 1-phosphate, which is converted to glucose 6-phosphate, which then can enter glycolysis in the cell. Liver cells, as well as some kidney and intestinal cells, also contain the enzyme glucose 6-phosphatase that removes the terminal phosphate, producing free glucose. Figure 2413 shows glycogenesis, the formation of glycogen from glucose, as well as the reverse reaction, glycogenolysis, the catabolism of glycogen to glucose. Glycogenesis occurs when glucose supply exceeds ATP demands. Extracellular glucose moves into the cell and is phosphorylated by ATP and hexokinase into glucose 6-phosphate. It then transforms into an isomer, glucose 1-phosphate. The terminal phosphate group is cleaved off, and the glycogen synthase catalyzes the product into glycogen. Liver and skeletal muscle cells are most active in glycogenesis and storage. On the other hand, when blood glucose levels drop, glycogen splitting occurs in a process called glycogenolysis. Lysis meaning splitting. So the splitting of glycogen. This is shown on the right side of the illustration. Glycogen phosphorylase phosphorylates glycogen into glucose 1-phosphate, which transforms into its isomer glucose 6-phosphate. Glucose 6-phosphatase in the liver, kidney, and interstitial cells remove the terminal phosphate, producing free glucose. Because glucose can readily diffuse from the cell into the blood, the liver can use its glycogen stores to provide blood sugar to other organs when the blood glucose level drops. Gluconeogenesis is the process of forming new neoglucose from non-carbohydrate sources. This occurs in the liver. Glucose can be formed from glycerol and amino acids when blood glucose levels drop.